All right, everyone, you know what they say. We save the best until last. I'm Robert Uhl from the San Diego office of Westwick Partners. As many of you know, San Diego is one of the leading biotech and pharmaceutical research centers in the world, and it's a pleasure to welcome a company based in San Diego as our final presenting company today. It will be Biomatrica, and the executive from the company to tell us about their exciting programs is Rolf Muller, who is President and Chief Scientific Officer. So I will turn it over to you, sir.
So thank you very much for still being here. And Rolf Muller, so I founded this company 7 years ago, and I don't have to tell you that cell-based therapy is important. But there's always a problem associated with it because all biological material degrade at room temperature. And it really impacts a lot of different areas, research development, patients, treatments. So I founded this company because we can make a difference by developing completely ambient biopreservation. So you know that very well. Small molecule drugs are very successful because they have a high stability, easy to use, easy to distribute. This is kind of out of scale because it's very difficult to produce because they have very low stability and very difficult to distribute. This completely goes and changes the value chain of a whole industry. So what is it? What actually makes it difficult? It's complicated. It's the most fragile drugs humans ever tried to evolve, ever tried to develop. And so Biomatrica, for the last 7 years, we developed biostabilization technologies, revolutionizing actually everything from forensic science to biobanking, working with 20 diagnostic companies in order to stabilize biopreservation material, the kits, biokits. So when you look at it, why do we think we can actually stabilize live cells? You look at nature, and there are plenty of examples where animals can survive in a dry state for a long time. Below, it's a tardigrade for 100 years in the dry state. You can resurrect these animals. So we have developed a technology platform to very quickly stabilize biomolecules, and last year we were able to actually preserve biological material at ambient temperature on the live cell level. So you can transport those cells around like normal small molecules. So how are we doing it? So it's a dry down technology. Cells which are in a growing culture can be just stabilized with a reagent. These biomolecules integrate into the membrane, are totally reversible, and leave after rehydration again. So we can dry them down, keep these cells in a stasis. At that point, they're completely viable, almost no metabolism, and a long-term preservation can start. With a rehydration buffer, we can bring them back to life, and most of the cells, after rehydration, start proliferating after 3 hours. So we have tested this technology in many different cell lines, primary cells, as well as stem cells. And here just an example. These are fibroblasts stored for 4 weeks in the dry state at ambient temperature, no cryopreservation, and we have about 95% survival rate at that state. Same thing, mesenchymal stem cells, and we look they behave completely normal. We did a lot of cell-based assays in order to test those things. Applications are very wide, and you know how these things can be applied. This is from wound healing down to improved drug discovery. So we have chosen cells that are very relevant to you in this kind of area when you go into keratinocytes, fibroblasts for wound healing repair, islet cells for diabetes, or glioblastoma cells, which we have for neuronal disorder repair. So regenerative medicine can be enabled by completely ambient biopreserved cells. So we are now going into the first animal model where we have 3 component cells. So this is mesenchymal stem cells, fibroblast, and keratinocyte dried down together, stabilized in a dry state. So you can then just ship them around, and after rehydration, we'll go into implants in order to see that we have wound repair similar to what normal stem cells can do. So what we are doing now, we have the technology. I'm now in the space of, so my other part of the company is fully funded. So now I'm going to find the right partner. So I will do it in a partnering model, the right partners, the right financing, pull it all together. And I think we can enable cell-based therapy like what we do with small molecules. So I think if anybody is interested to work with me on certain approaches, this is exactly the right time to push this technology into development. And I think a lot of people don't need to go into cryopreservation, have liquid nitrogen tanks being distributed around Australia, for example. So thank you very much for your interest, and I think we can have a drink together and to talk about what we can do. Thanks.